Hello friends. So far we have discussed about the structure of the carpel and the number of the carpels or the types of the carpel depending upon what is the number present, right? So now we will be studying about the most important part, that is the ovule, or which can also be called as the megasporangium. So we'll be seeing the structure of the ovule inside which the female gamete is going to develop. So basically, your ovule inside the ovary is going to appear somewhat like this. We will study what are the different parts, and then we will move ahead further to see what are the functions of each and every part. Okay. Now, you must have studied about placentation in 11th standard, right? So if I take the example of marginal presentation, you know that marginal placentation is found in the pea. So you know that within a pea pod there will be a small connecting stalk-like structure to which the seed or the ovule will be attached. Of course, this ovule is later on going to be called as the seed. So how these ovule is attached with the help of the placenta to the mother's body, or when it is present in the flower in the ovary, that is the structure that we'll see here right now. So when I say that this part, the stalk which is attaching the ovule to the placenta is called as funicle. What is the function of funicle? Stalk that attaches ovule to placenta. So if I want to say, I can say that here will be the placental tissue to which this ovule is attached. Fine. So the stalk which is actually connecting both of them, that stalk is called as the funicle. The funicle is going to further extend and connect with the structure of the ovule. So that point of the ovule, or you can say that point where the funicle and the body of the ovule, they are fusing together, that structure or that part is called as the hilum. Funicle is the stalk which connects the ovule to the placenta. Hilum is that part where you can say funicle fuses with body of ovule. So this is the common part between the funicle and the ovule. Now, if you see the complete ovule, you will see that it is protected by many layers, right? So these layers, they are actually called as integuments. This will be called as the outer integument, and this is the inner integument. There are almost 2 to 3 layers of integuments that are present, which are acting as the protective layers of the ovule. So their function will be, they are protective layers. Next, inside the integuments, there is a region where the mass of cells will be present, and that region is called as the nucellus. Okay, nucellus is the mass of cells that is protected by integuments. Now actually one of the cell of the nucellus is going to give rise to the megaspore mother cell from which the female gamete will be developing. We will study about that later. Let us first see about only the structure of the ovule. Now if you see clearly the structure, you will see that these integuments, they are protecting or covering the entire surface of the ovule except at one point where there is a small opening present. That opening is called as the micropyle. That opening is called as the micropylar pore. It can be called as a micropyle end as well. So what is the function of these micropyle basically? It is the end through which pollen tube enters inside the ovule. So you can say it is the entry point for the pollen tube. Right? Now exactly opposite to the micropyle, the one end is present. That end is called as the chalazal end, or it can be called as a chalaza. It is present, you can say, the end opposite to micropyle end. So these are the different parts of the ovule. Now inside this ovule, as I said, will develop the embryo sac, or you can say the female gametophyte, right? And these embryo sac is actually going to develop from one of the cells of the nucellus. That will be coming under the process of megasporogenesis. So let us just see what are the different structures of the ovule. Funicle is the stalk which attaches the ovule to the placenta. Hilum is that region where the funicle and the body of the ovule, they are fusing together. Micropyle is the end where integuments are absent and the point from where the pollen tube will enter inside the ovule. The protective layers present inside the covering the ovule are called as the integuments. There are almost 2 to 3 layers and their function is protection. Inner to the integuments there will be a mass of cells which is called as nucellus, from which will develop the female gametophyte or the embryo sac. And the end exactly opposite to the micropyle end is called as the chalazal end. Now, this is the most common type of ovule, or you can say the anatropous ovule, which is found in angiosperms. Depending upon the arrangement of the funicle, micropyle and the chalaza, what is the positions of these three? Funicle, micropyle and chalaza. Depending upon that, there are 6 different types of ovules found in different species of plant. So let us now see what are the types of ovules. Okay, the first type of ovule is orthotropous ovule. Now in case of orthotropous ovule, what you see is orthotropous ovule, what you see is the funicle. This is the funicle, this is the mitropyle, and this is the chalaza. So you can say that the chalaza, funicle and the mitropyle, they are all in the same line. So these type of ovule is also called as the upright ovule, where the mitropyle, chalaza and the funicle, they all fall in a same line. It is mostly found in gymnosperms. This is the most common form of ovule that is found actually in gymnosperms. The second type of ovule is called as anatropous ovule. Now these ovule is actually the inverted ovule, the one which we saw just now. It was an inverted ovule. This is the funicle, this is the chalaza, and this is the mitropyle. So what you can say that the chalaza and the mitropyle, they are present at the right angle to the funicle. They are not exactly right angle, but the arrangement is such that the chalaza and mitropyle, they will come in a common line. And this is the most common type of ovule in angiospermic plants. That means whenever we talk about the angiospermic ovule, we will talk about the anatropous ovule. The third type of ovule is called as hemitropous. Now in case of hemitropous ovule, what happens is, so this is your embryo sac. This is the chalazal end, this is the mitropylar end, and this is the funicle, where the chalaza and the mitropyle will be completely inverted at right angle to the funicle. So these type of ovule is called as the hemitropous ovule. The fourth type of ovule is called as campylotropous. In case of campylotropous, what you see is, if you see this, it is completely inverted in case of anatropous ovule, and here it is at right angle. So neither it will be completely inverted nor it will be completely at 180 degrees Celsius, right? Sorry, 180 degrees. So what happens? There will be certain angle at which the ovule is present. So this is the mitropyle, this is the chalaza, and this is the funicle, and inside is the embryo sac, right? So this, see, it is neither completely inverted like it is in this, neither it is completely in the 180 degrees. So it is somewhere in between. It is more than 90 degrees and less than 180 degrees. That type of ovule is called as campylotropous ovule. The next type of ovule is called as amphitropous. In case of amphitropous ovule, the angle of these three, funicle, chalaza and the mitropyle, will be same. That means it will be somewhere present at an angle. But apart from that, if you see here, the embryo sac is also present at an angle, or you can say its shape has been curved. Or you can say in the amphitropous ovule, the embryo sac is horseshoe shaped. Of course, the angle of the funicle, chalaza and the mitropyle will be same as it is in Campylotropis, but here the embryo sac is intact and it is not present at an angle. Whereas here the in case of Amphitropis ovule, the embryo sac is also curved in horseshoe shape. The last type of ovule is Circinotropis ovule. In case of Circinotropis ovule, what happens is the funicle has extended so long that you can say that the funicle itself this is the embryo sac, chalaza end, mitropylar end and the funicle end. So if you see here, it will be same as that of the Anatropis, where the chalaza and mitropyle, they are in a common line and they are opposite to the funicle. But that inversion of the ovule is because the funicle has extended to a very large extent and they are going to somewhere fuse with, you cannot clearly distinguish between the point from where the integuments are going to start. So this type is called as the Circinotropis ovule. These are the 6 types of ovule which are found in different species, among which you will see that the most common in Gymnosperm is Orthotropis and the most common in Angiosperms is Anatropis. There are certain exceptions in certain species of plants in which you will find this type of ovules. So this is all about the structure of the ovule, or you can say Megasporangium, and what are its types. Thank you.